Howdy, my name is Sarah Wines, and today I will be presenting my research project on the genetic and chemical triggers of the variosensitive hygiene response in Apis mellifera. So as a background, Apis mellifera are also commonly known as your Western European honeybee. They are extremely important for the agricultural economy and are one of the best pollinators that we see on Earth. They are actually related to Apis cerana and migrated from Asia to the Western Hemisphere, where we will be focusing on the United States population. So these insects are actually extremely sensitive to their abiotic and biotic factors around them. One of the most important and uprising threats that Apis mellifera is currently facing are pests. One of these pests is actually an ectoparasite called variodestructor mites. Their original parasitic host was the Apis cerana, and these mites are actually responsible for mass decimation of bee colony health due to their viruses and their fast, large infestation rates. So as a result of just have been lots of effort put into researching treatments and responses by Apis mellifera, one of the natural characteristics that scientists have found is the variosensitive response, hygiene response, which is similar to hygienic behaviors that we know, such as grooming, that we see in the bees' social behaviors. So the VSH response has actually been known to have genetic and chemical triggers when associated with mite presence in Apis mellifera colonies. As a visual, on our left, we have a variodestructor mite on the abdomen of a removed Apis mellifera pupa. So this pupa was removed before maturation. On the right, we have a worker bee of an Apis mellifera, which is what you'll see when you think of a nurse bee or any female bee besides the queen. So the specific question I am asking are, what are some genetic and chemical triggers of the variosensitive hygiene response in Apis mellifera when they are in the presence of variodestructor mites? One of the first genetic factors that we have found is actually the differential gene expression that correlates to their proteins. So in this experiment, they tested, and I quote, "Thus here we tested the hypothesis that intercolony variation in disease resistance parameters is reflected by changes in the expression of specific proteins." So what they did is they took samples from 40 genetically different colonies and took antennal samples from their nurse bee populations and the skin of, or the chitin of, the fifth instar larvae in these colonies as well, and chemically analyzed them through proteolytic digestion, which is just the breakdown of these samples into their specific protein units. And they found a positive and negative correlation with the VSH response and the proteins that were expressed in these samples. So on our top diagram, we have a heat based assay, which shows positive correlations in the green and negative correlations in the red. On the bottom diagram, we see a positive correlation with the signaling pathways and a negative correlation with the carbohydrate metabolic processes. So pretty much in general and just in overall, what they found with this experiment was that the differential gene expressions and their rates affected the rate of the VSH response, which we normally see with these proteins that they found is the differences in the inflammatory response and chitin production and chemoreception of the neural pathways. One of the other triggers is developmentally delayed pupa. So the VSH response is actually seen when nurse bees will take and uncap a capped brood cell and either remove the pupa or recap it to disrupt mite reproduction cycles. So the theory or not the theory, but the hypothesis that this experiment was testing, and I quote, "Our findings suggest two testable hypotheses. First, that VSH is triggered by unusual pupal BEP profiles, specifically associated with vario-infested brood, and second, that these altered BEP profiles are due to developmental delays caused by specific vario-transmitted virus infections." So what they're essentially saying is that developmentally delayed pupa are removed and trigger the VSH response because of their viruses that they've gotten. So what they did is they took an infected brood cell and they transplanted it into a colony that has VSH responses observed already. And after an hour, they took it to the laboratory, tested it, brought it back, and left it for another hour until 48 hours of observation have passed. And there they decided to observe and correlate the amount of brood and mite content in their cap cells and their surrounding cells and dictate their stages, as well as do a virus titer in the capped brood cells to determine the amount of targeted cells that had the specific virus is present. So what they found is that the brood cells that were targeted compared to their surrounding cells were developmentally delayed between 3 to 5 stages between their control groups. And with the viruses, they found that there was a higher presentation in one virus versus the second virus, but that the first virus shows that they weren't detected by the VSH response, but the other was. So what we can kind of garner from this experiment or this hypothesis was that the VSH response does go with developmentally delayed pupa, but it may or may not be based off of the viruses themselves. The last chemical trigger that I found was an excess of brood ester pheromone. So this was about the first part of their hypothesis. It was the same setup, except instead of finding their viruses and comparing them to their neighbors, they did a quantitative analysis of their brood ester pheromone profiles. So from the diagram, the targeted cells, which is also marked as TA, were known to have a large variation in their brood ester pheromones. In fact, they found a 130% increase from these targeted cells compared to the control groups of the pheromones that they emitted. From these pheromone emitted, the VSH response was triggered because they were removed from their cells. In conclusion, the variosensitive hydrogen response is brought on by genetic and chemical triggers. Specifically, the different proteins expressed in the populations can dictate whether or not the response is given based off of correlation. Additionally, the production of an abnormal amount of brood ester pheromones in brood cells triggers the response of nurse bees to remove the afflicted pupa. Not to mention the developmentally delayed prepupa and pupa from infected brood cells also have the diseases that are transmitted by the mites. So the significance is by fully understanding what triggers the variosensitive hydrogen response, scientists can kind of start to develop a more efficient rate of reaction so that Apis mellifera have a better chance of reducing these mite populations naturally. Now that we know that VSH is correlated to differential gene expression, beekeepers can start to look at breeding their queens to drones who are known for these different genetic expressions so that there's a higher response in future generations. And if scientists can find what controls the response, it can be manipulated for the benefit of honeybee populations without truly interfering with external treatment options. These are my works cited. And thank you very much.